my name is Jiya Anand. I'm a junior at Marjorie Stoneman Douglas High School in Parkland, Florida. And today I'll be presenting the Quantitative Regional Differences in Autism Spectrum Disorder, distinguished from controls using the ABIDE2 database. So, autism spectrum disorder is a neurological disorder that is caused by genetics, which affects the development and social behavior of a child. For example, a variety of symptoms include impaired social communication, interaction, and repetitive behaviors. While a root cause or cure hasn't been found yet, it is known that the earlier you diagnose ASD, the easier it is to manage. Therefore, by utilizing the structural MRI neuroimaging technique, we hope to be able to pinpoint the exact location that autism is making its mark on, and then interpret what effects are present mentally, physically, or emotionally due to the change in the size of the brain. In order to be able to further dive deeper on ASD, it was important to understand the already known information on factors known as, such as locations where physical makeup is known to be affected and how age plays a role in the anatomy of the brain. There are three main areas that are well known for overgrowth and being disordered in ASD, which are the amygdala, the orbital frontal cortex, or the OFC, and the temporal parietal cortex, or the TPC. These areas will often see the most drastic impacts with both the physical and the internal makeup of the location. Overall, all three of these are generally responsible for tasks that have to do with social capabilities and the way you uphold yourself in front of a person, such as body posture or behaviors that are used when presenting yourself to another human. Age is a factor when analyzing total growth and change of the physical structure of the brain. As we see by the image, with children with ASD, there is abnormal brain growth early on, as well as abnormal brain volume decline later on at around age 15, compared to a typical control, which is significant when analyzing the development of the brain. So, our data was sourced from the Autism Brain Imaging Data Exchange, or the ABIDE2 database. ABIDE2 is a collection of 16 international neuroimaging sites that openly shares neuroimaging data from 463 individuals who suffer from ASD, as well as 541 healthy individuals. In the ABIDE database, both the healthy controls and the ASD group had a mean age of 15, so age was not considered during this study. So to go further in depth, we decided it would be best to study specific locations in order to analyze quantitative differences in surface area, volume, gyrification, and cortical thickness to see where exactly the changes are in order to decide if those areas are heavily impacted. So by conducting a t-test in order to distinguish which areas vary the most between ASD and typical development, we were able to find the top three regions of the brain that presented to be the most different. We then used those significant regions to try and link and find a correlation in anatomy to the Autism Diagnostic Observation Schedule, or the AUDOS, and the Autism Diagnostic Interview, or the AUDI, which are autism spectrum disorder measuring techniques, which determine the level of development a child has reached. So, after conducting a t-test, the top three regions of the brain whose surface area were most affected were the left OFC, where the typical development group has a 7% increase in average surface area. The right OFC has a 6% increase, and the right superior temporal sulcus ventral posterior, or the STSVP, has a 5% increase. In terms of volume, where areas were most affected, the left STSVP showed that the typical development group has a 7% increase in total volume. The right STSVP has a 6% increase, and the PHA1, or the parahippocampal area 1, has a 5% increase. For total gyrification, in the left OFC, a 7% increase is present, and in the right OFC, a 6% increase is shown, and in the right STSVP, a 5% increase in the typical development group compared to the ASD group is shown. Finally, for cortical thickness, there seems to be a 5% increase in the typical development group than the ASD group, and which is also known as the frontal opercular area 1, or the FOP1. A 3% decrease in the typical development group is shown compared to the OFC group in the OFC. And a 3% increase in the typical development group compared to the ASD group is shown in the 47's lateral orbital sulcus, or the A47S. So, after conducting a correlation between each of the 12 measures that I just discussed, we were able, and AUTOS and AUDI, a null linear correlation was present. We suggest that this could be due to the fact that these measures are an indicator of whether or not, or whether that the clinical presentation in the brain. Although there is a difference physically in the brain, the typical development in the ASD group, in the typical development in the ASD group, we hypothesize that AUTOS and AUDI are attached to internal clinical factors rather than the anatomical presentation of the brain. So, and then some of the limitations that we were able to find in our study were the lack of consideration with age. Although the growth of the brain is definitely impacted by the age that a person is, because the ABI database had a mean age of 15, we did not take that into account, as well as inconsistencies with the MRI data. Now, the ABI database has been around since 2007, so inconsistencies will happen to occur due to the amount of changes that have been made since then. Finally, exclusion of other regions. There are many other regions in the brain that are physically affected by autism. However, though we only sifted out the most important and significant regions to make our presentation today. In conclusion, findings from this research study confirm and support findings of other existing studies that focus on morphometric abnormalities in the anatomical makeup of the brain. And we hope that this study can contribute to further research in the future that lead to the root causes of ASD and quicker diagnosis in order to start treating at a younger age. Thank you.
Amazing job. All right, we have one question for you. Let's see here. What are the next steps for you now that you've completed your project? Like, will you continue to build on this?
Yes. So, like I stated, we definitely took an overall approach. Like, we looked at totals, and it was more general. So, for like an upcoming project that I was brainstorming, I hope to go further into about gender specifically, and how, because autism is definitely more prevalent in males rather than females. And so there are larger changes in males and females. So I want to go more into depth about the physicality of the brain, but in gender.
Very cool. Can't wait to see what you learned in that.
Thank you.
All right, great work.